Our group decided to do the blood typing experiment, and this is a presentation of our report. The purpose of this experiment was to determine a patient's blood type in order to do a safe blood transfusion. In this experiment, at-home materials were used to simulate how blood types are identified, which is done by taking a blood sample from the patient, in this case milk, and testing it against different anti-sera, which was water mixed with vinegar. Our results concluded the sample was type A. In blood typing, the blood is tested for the presence of A and B antigens, as well as the Rh factor, and the blood type is determined by the combination of these proteins on the surface of the red blood cells. It's important that during blood transfusions, a compatible donor is used, as an incompatible blood type triggers an immune response in the body, where the body attacks the new red blood cells and destroys them.
Methodology. Using the whiteboard markers, the 4 cups were labeled consecutively. Cup 1 reading blood, Cup 2 reading antiserum A, and Cup 3 reading antiserum B, Cup 4 reading anti-D Rh serum. The marker was used to label 3 small bowls as follows. Cup 1 blood plus anti-A, 2 blood anti-B, 3 blood plus anti-D. In the cup labeled blood, a quarter cup milk was added. 20 drops red food coloring was then added. Stirring was done in between each addition. This was repeated until the color resembled a blood-like hue. In each cup labeled antiserum, a quarter cup water was added. 1 tablespoon of vinegar was added into cup B. Another person should add 1 to 1.5 tablespoons of vinegar to 1 or 2 of the cups of antiserum. 1 spoon was placed in each cup of antiserum, and cross-contamination was prevented by not using the spoon for anything but mixing with antiserum. In each small bowl, 2 spoonfuls of blood hue was added. The small bowls and cups were lined up so that they were readable. In the bowl labeled blood plus anti-A, 1 spoonful of anti-A serum was added and mixed. In the bowl labeled blood and anti-B, 1 spoonful of anti-B serum was added and mixed. In the bowl labeled blood plus anti-D-RH, 1 spoonful of anti-D-RH serum was added and mixed in. Each bowl's reaction was then observed.
The material that we use is seen in figure 4 materials. It was a cow's milk, water, and the vinegar and the red food coloring, permanent marker, and the tape, 3 bowls that we used there, and 4 cups, and the 1 tablespoon, and 3 stirring rods.
The aim of this experiment was to determine the hypothetical blood type of the sample of blood. As seen in the table and the image of the observations, clumping was evident in bowl 1 but not in bowls 2 or 3. This result indicated that the blood type of the sample was A negative. Moving on to the discussion. A possible limitation of this experiment is that of human error. If the spoons placed in the various bowls were used in any other bowls other than that in which they were originally placed, cross-contamination may have occurred. This obviously would have altered the outcome of the experiment. Having a look at the diagram on the right, it indicates that the universal donor blood type is O negative. This means that blood type O negative can be a donor to any other blood type. A person with blood type A negative, as in our case, can only donate to others of blood types A negative, A positive, AB negative, or AB positive, and receive blood of types O negative or A negative. Since the blood type is A negative, it contains antigen A and does not possess the Rh factor.
Our recommendations were to repeat the experiment by adding vinegar to different bowls to obtain different outcomes. The control measures prevent the contamination by not using the same stirring rod in different bowls. We recommend using clear containers so that the results are more visible to be seen. Figure 7, it is a table that shows the possible outcomes that had been with the vinegar in different bowls. However, in our experiment, the blood type was determined as A. This means the donor required the blood from A only, or negative, or O negative. The blood type is determined on agglutination patterns. This is